Hi everyone, and welcome back to another episode of Science. In this episode, we'll be having a look at a biological process called mitosis. It's one of my favorite areas of biology, and I'll explain why in this episode. So let's get started. Mitosis is the process that your body uses in order to grow, repair, and replace its damaged or dead cells. Your body is made out of trillions upon trillions upon trillions of cells, and every second, millions of those cells die. But luckily for us, through the power of mitosis, your body is able to also make millions upon millions of new cells every second. Now, your body is undergoing mitosis right now in order to grow and repair itself. In this particular slide, I want to go through how mitosis allowed you to go from this single cell to being born as a baby. Believe it or not, all of us looked like this at the very beginning of our life, as an embryo. Once the sperm fertilized the egg, and this is indeed an egg cell, mitosis could now begin. Now, mitosis allowed this one cell to duplicate into 2 cells, into 4, into 8, and so on and so forth until you were born as a baby that looked probably something like this. The question is, how does your body do this? How does it go from 1 cell to 2 cells to 4, all the way up until you were born as a baby, and all the way up until you watching this episode of Science right now, and all the way to adulthood, and all the way to death when mitosis will stop? Well, we need to have a look at what's actually happening inside of the cell. Firstly, what the cell needs to do is double all of its material, so all of its organelles, the machinery of the cell, and also the DNA of the cell. It has to double it all in order to divide and 1 cell go into 2. We'll have a look at that DNA duplication process in this episode. Now, what also needs to happen is that the cells need to become a little bit more specialized. What you'll notice in these diagrams is that these cells look the same. They are identical to each other. And they are indeed identical, and they contain the same DNA in each of them. All of your cells actually contain the same DNA within their nucleus, no matter what type of cell you're talking about. But what you'll also notice is that this baby has got specialized skin cells and heart cells and eye cells and all the cells that allow it to live, all the tissues that allow it to live. How does your cell go from 1 cell into being born as a baby with all these unique specialized cells that allow for the specialized functions within your body? Well, within the nucleus of each cell, you'll find your DNA, and that DNA has got the instructions. However, depending on what your body needs, depending on the developmental process, different sections of the DNA will be turned on and other sections will be turned off. Now, how your body knows which sections to turn on and which sections to turn off is still an area of study within biology. So if you're curious about that, maybe VCE biology and even further studies beyond that could interest you. But the genes, which are the instructions found in your DNA, get switched on and off. So this particular picture here, in this example, the genes that tell your body to grow your vascular system, your veins and arteries, have been switched on, and they are growing throughout your body. Also, the genes to grow eyes, the genes to grow ears, arms and legs are all switched on. And when they are fully developed, some of those genes are switched off. It just depends what your body needs at the time in order to grow and repair itself. At the end of this process, assuming everything has gone according to plan, of course we know not everything always goes according to plan, you're born as a healthy baby with all the functioning and specialized tissues you need in order to survive. But it's the process of mitosis that allows this to occur. All of us went from one cell to being born as a baby. That's pretty amazing in my opinion. Let's talk more specifically about the DNA though. How does the DNA duplicate? How does the DNA multiply in order to go from one cell to two cells? Let's have a look at that. In this particular diagram, I've got my original double helix strand of DNA. And this is the strand of DNA that I need to duplicate. I need to double my genetic material. And so a special enzyme called helicase will come along and it will unzip your DNA double helix strand, similar to like a zip on a jacket. The helicase will come along and unzip your DNA strand. One strand will go up here, and another strand will go down here. And now you've separated your double helix into two separate strands. From here, another enzyme will come along, and that's DNA polymerase. The job of DNA polymerase is to grab some free-floating nucleotides that you find in your nucleus. Your cell produces nucleotides to be used when they are needed. Your DNA polymerase will grab the right nucleotide and bond it to its complementary nucleotide. And if you think back to last episode, A always pairs with T and C always pairs with G. And the DNA polymerase will make sure that you've got the correct complementary base pairing rule being followed, and it'll do it all the way to the end of your DNA strand, and you'll end up with two new DNA strands that has come from the original DNA strand. This one will go to one cell, and this one will go to another cell. Now, on this slide here, we've got all the DNA that's found within your nucleus, keeping in mind that you have the same DNA in all of your nuclei. But different sections of the DNA are used depending on what cell we're talking about. So a skin cell will use different sections of your DNA code than your heart cell. Nonetheless though, all of your cells have the same code. It just depends on which genes are switched on and off that determines the function of the cell. This karyotype here is showing all the DNA within the nucleus of a cell. These sausage-looking things here are called chromosomes. We've mentioned these in the previous episode, but we'll go into a little bit more detail as to what a chromosome is. Simply put, they are a chunk of DNA. And you've got lots of chunks of DNA. Specifically, you've got 23 pairs of chunks, giving you a total of 46 chromosomes. So this is the first pair, second pair, third pair, all the way up to the 23rd pair. Your 23rd pair are actually your sex chromosomes. So for this particular human, I have a Y chromosome and X chromosome. This makes this person a male, a boy. Girls have two X chromosomes. So this one's an X and this one is a Y. You'll notice the Y chromosome is much smaller than the X chromosome, and that's because there's less DNA in the Y chromosome. There's less instructions, less genes in the Y chromosome compared to the X chromosome. You'll also notice that they come in pairs and they're the same shape and the same size, and that's because they are what's called a homologous pair. Homologous means the same. So what do we mean by the same pair of chromosome? Well, one chromosome of a pair comes from mum, and the other chromosome comes from dad. So you inherit one set from your father and one set from your mother. So if I was to draw here a sperm cell and the larger egg cell, your mother's egg cell has 23 chromosomes and your father's sperm cell has 23 chromosomes. And when the sperm fertilizes the egg, you end up with 23 plus 23, giving you a total of, I'll just draw it here, 46. And so this is where all of your chromosomes come from. One is from dad and one is from mother. One is from the sperm and one is from the egg. If I come back to this slide for a moment, so this egg cell has got 23 chromosome, and I believe this arrow is pointing to a sperm cell, but it's much smaller, so you can't quite see it in this image. But the sperm cell has 23, and once fertilization happens, each cell after that will have a total of 46 chromosomes, which represents all of the genetic material within the nucleus of each cell. So that's why we say 23 pairs. 23 pairs gives you a total of 46. Now, another interesting thing about these chromosomes is because they're homologous, the same, the instructions on them are also matching. So, for example, I'll just do a line here. This section of chromosome pair number 1 could represent hair color as an example. So you inherit instructions from your father for hair color and your mother for hair color. Sometimes the gene from your father is stronger than the mother, and so your hair may look like your father's. Or sometimes the gene from your mother is stronger than your father, so your hair may look more like your mother's. Or sometimes the genes are equally strong to each other, and so your hair will look like somewhere in between your mother and your father. Perhaps your father has black hair and your mother has blonde hair. You may end up with something in between, brown hair. It depends on what characteristics, what trait you're talking about. But for each of the pairs, you'll find the same genes at the same at the same location. Same genes at the same location. And sometimes one gene is stronger than the other. And this is why you can sometimes look a little bit more like your father or your mother or something in between. And we refer to these pairs as a homologous pair. You get one from dad, one from mum. Let's continue. Let's have a closer look of how these chromosomes actually need to multiply in order for your cell to divide, in order for your cell to undergo mitosis. So here I'm taking one homologous pair. So let's just pretend I take this first homologous pair, keeping in mind you've got 23 pairs, but I'll just take one pair and put it here. So here I've got one homologous pair. This one we'll say belongs to your dad, and this one belongs to mum. So you've inherited this chromosome from your father's sperm and this chromosome from your mother's egg. You need to multiply all of your chromosomes. You need to double them all up. So what ends up happening is, here is the original chromosome from dad. Your cell needs to make a copy of it, and so we can make this one the copy. Here is your mum's original chromosome, and this one can represent the copy of the chromosome. So here, if I take into consideration all of the chromosomes at the very beginning, I have my standard 46, 23 from dad, 23 from mum, making a total of 46. But once I have duplicated all of the pairs, all of the chromosomes, I end up with a total of 92 chromosomes. And these duplicated chromosomes, we call them sister chromatids. Sister chromatids. So this one is a copy of this one, and this one is a copy of this one, and we call them sister chromatids. They're joined in the middle by a structure called the centromere, and that's to keep things orderly. So what happens after we've multiplied our DNA? What's the next step? Well, the next step is to divide the cell itself. So let's have a look at this process. You can use an acronym to help you remember this process. So IPMAT here. Now the I represents interphase. And interphase is where your cell, where your nucleus duplicates all of the chromosomes. So this is where the duplication is occurring. During prophase, that's when all the DNA strands, all the chromosomes condense and become visible. So you can see them under the microscope. Usually they are too small to be seen even under a microscope. A normal microscope, that is. You can use an electron microscope to get a grainy image of them, like we can see here. So this is an electron microscope. Obviously it doesn't give you a very good resolution. But during prophase, they condense and you can see them under the microscope. These here now have taken on their classic X shape. We normally see chromosomes depicted as this X shape, but if you see them as this X shape, it means that they have duplicated. Normally, they should look like this. They are separated, one's from dad, one's from mum, and then they take on the X, the famous X shape, only after they have duplicated. So multiplication occurs, the chromosomes, they condense, and during metaphase, the chromosomes will line up along the equator, like so, and these structures called spindle fibers will come out from the centrosome, connect to the centromere of the chromosome. So those spindle fibers connect to this structure here, and the spindle fibers will begin to pull apart the chromosomes. And so here you've got your 92 chromosomes. Here you've still got 92 because everything's duplicated. But here, this is when the spindle fibers separate the chromosomes. So your 92 now returns to the normal 46 for one cell and 46 for the other cell. You'll also notice that the cell is starting to pinch in the middle. And the next step, telophase, is when the chromosomes have been totally pulled apart and the cell is about to totally pinch in the middle, where one cell will turn into two cells. You'll also notice that there is a new nucleus surrounding these chromosomes. Between the interphase, prophase stage, the nucleus actually dissolves and goes away. And then between the anaphase and telophase phases, you develop two new nuclei that encases the genetic material of your two new cells. So here we've also got 46, 46. And chances are these two cells will further go on to undergo mitosis. So they'll double up their genetic material again, and then they'll split as well. Use this acronym to help you remember the phases: interphase, prophase, metaphase, anaphase, telophase, or IPMAT. I've also got a couple of videos for you to watch here, so some real microscopic mitosis occurring here. Definitely recommend you have a watch of that. But from here, I would recommend going back and having a look at this PowerPoint, have a look through your textbook. Education Perfect has some good resources. But brings us to the end of this episode. Thank you so much. I hope you enjoyed this episode, and I'll see you next time in class.